Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today. Treatments for HIV are a crowning achievement of modern medicine. A patient living with HIV in the Western world can now expect to live as long as those without the virus. Yet a cure has proved elusive so far. Why is this the case? Could we hope for a cure in the future, and where could we look for it? To help answer these questions, we're joined today by Professor Andrew Lever, an honorary consultant at Addenbrooke's Hospital in Cambridge and Professor of Infectious Disease at Cambridge University.
I think if you ask anyone with HIV whether they'd like something simple and curative rather than taking drugs for the rest of their life, they would choose the former. And of course, it's very well controlled, but it's still a large drugs bill. The estimate for worldwide anti-HIV therapy for 2020 is that it's going to cost $25 billion or something like that. So it's a very large health burden, particularly affecting countries where health resources are relatively limited. So anything which gave a reliable cure and prevented both the individual having to take drugs, but also the government have to pay out to supply all these drugs, would be a very welcome occurrence indeed.
Although perhaps, to be slightly cynical, the pharmaceutical industry has often been criticised, and certainly lots of criticism has come out recently, for strategies that favour treating chronic diseases with, for example, daily treatment that's quite lucrative, and actually shying away from potential cures. Is there an issue here that actually a cure for HIV for the very companies that perhaps would be in a very good position to make it aren't necessarily the marketing strategy they'd like?
It's possible, but I don't see people actively trying to avoid developing a cure. And again, without being the shining knight defending the pharmaceutical industry, what they've achieved so far has been really very, very dramatic indeed.
Yeah, of course.
So I think it's more an issue that a cure is so incredibly difficult, and the way towards a cure is not really obvious as yet, rather than people deliberately avoiding trying to find out how to cure HIV.
So why exactly is HIV difficult to cure then, compared to, say, other viral infections?
So there are a number of issues here. Curing AIDS is obviously much easier because you can reverse all the damage the virus does, but you can't eradicate the virus. But one has to remember that it's not the only virus that is difficult to eradicate. We're all infected with a number of herpes viruses, herpes simplex, varicella zoster, the chickenpox virus, cytomegalovirus. And once you've got those, you never lose them. We've never been able to eliminate those viruses. And these share the characteristic of HIV that they're chronic viral infections. And that distinguishes them from things like measles and mumps and whatever, which is a transient infection which infects somebody and then goes on to infect somebody else. And once you've had it, your immune system can cope with it and you don't get infected again under normal circumstances. And those viruses have that strategy for surviving, of finding new victims as fast as possible. Chronic virus infections in general have got to survive a long time so that they can take advantage of the relatively few opportunities they get to transmit from person to person. And as such, they all have strategies to avoid or hide from the immune response, or to suppress it one way or the other, so that they can last a long time in their infected host and get an opportunity to transmit to somebody else. So it's a simple case of, if you're a chronic virus infection, you're rather good at hiding away from the body's defences. And HIV has a very significant number of capabilities of doing that, as do other viruses.
So what sort of things are these capabilities of hiding from the immune system?
Well, one of the things I've mentioned already, which is the ability of the virus to mutate and change its coat so that the immune system no longer recognises it. And that's extraordinarily effective. It also, of course, depletes the immune system itself, which makes the immune system, again, less able to eradicate it. Probably its most potent weapon, however, is that, again, in common with some herpes viruses, it's got this ability to go latent. So the virus can infect a cell, and as I mentioned, its DNA will integrate into the genome of the cell and then produce new viral RNA and viral proteins. But in some cases, in a proportion of cells, the cell will be undergoing a change in state. It will be itself changing, in the case of a lymphocyte, to a memory cell and shutting down some of its processes. And that seems to coincide with, if that's the time at which the virus infects the cell, the virus itself switching off and effectively going to sleep. So it's no longer producing any RNA or any protein. And that means all it is a piece of DNA which looks like any other gene and is not recognizable by the immune system. There are no viral proteins produced, so the immune system can't see it. And there's nothing to kill the cell either, so the cells should survive with this Trojan horse of a virus inside it. And these latent cells exist in everybody who's infected. They're, of course, extraordinarily difficult to identify because there's no viral signals that they're infected. And they hide away in various sites in the immune system as well, which some of which we know, but some we're still finding out.
I know there's a little bit of controversy over whether latency occurs in cells of the myeloid lineage. Do we have a clear picture roughly of which cells could be harbouring latent HIV infection?
So the problem population is almost certainly the CD4 positive lymphocytes, the so-called T memory cells. Because they get infected, they become memory cells, the virus goes latent, and these cells can survive for decades, which means that the virus is able to survive for decades and then can reactivate at a later time if the conditions are right. It's possible, although still not fully proven, that latent virus exists in cells of the monocyte macrophage series, but these in general tend not to be as long-lived. And circumstantial evidence suggests, perhaps from the few cure cases that have occurred, that this may not be quite as important a latent reservoir as the lymphocyte population.
So there have been a handful of cases so far in which patients are thought to have been cured of HIV. I know you've worked with the London patient, which is one of them. Could you tell me a bit about these?
So yes, probably two cures.
So slightly less than a handful.
And even those we don't absolutely definitely know, but they're looking very promising. So the first one was the so-called Berlin patient, and this was somebody who was HIV positive and was on suppressive therapy that then developed a malignancy, a hematological malignancy, and had 2 stem cell transplants. And the donor of the bone marrow transplant that they received was one of these unusual individuals who had a genetic abnormality in which they were missing one of the proteins on the surface of their lymphocytes, which is one of the receptors that the virus uses to get into cells. And about 1% of the Caucasian population in Europe are homozygous for this mutation, which means that they can be exposed to HIV, but the virus in general can't get into their cells. So the bone marrow this person received had a homozygous deletion for this protein, which meant that even if there had been any residual virus in the person who had the bone marrow transplant, it wouldn't have a bone marrow into which it could infect its genome. And that seems to have been the case, and that now is around 10 years down the line from the second bone marrow transplant this individual had, and they're still well with no detectable HIV in them.
And this was quite a radical treatment.
It's not a straightforward treatment.
And not designed to treat HIV, this was a byproduct.
It absolutely so. It's not something you put anybody through just to treat HIV, particularly when you've got such good treatment available to suppress the virus. The second case, the so-called London patient, is much more recent and had the bone marrow transplant just 2 or 3 years ago, and had their anti-HIV again with somebody who is CCR5 negative as a bone marrow donor. And again, it's been impossible to find virus in that individual since that time, and their antiretroviral drugs were stopped in 2017, and we've been involved in doing detailed assays to try and detect latent virus in this individual on 4 or 5 occasions now, and we cannot detect it at all. So the suspicion is that person may also be cured. We don't know for certain, because if there was a very, very tiny reservoir somewhere, it may be in T memory cells which have not had the opportunity to reactivate as yet. So ongoing monitoring is still going to be necessary for this individual. I don't know how long we'll have to do it for, but I suppose if it's gone on for 10 or 15 years, you'd say it's pretty unlikely you're still going to find a virus in them.
Yeah, I guess it's a difficult question, as absence of evidence doesn't necessarily mean evidence of absence.
That's true.
So given it's I guess, relatively low efficacy and high cost, and actually a lot of risk associated with it, stem cell transplantation doesn't necessarily seem to be that viable for mass treatment of HIV. Have any other treatments been suggested?
So you're quite right. And in fact, people get the bone marrow transplant, as you said, for the reasons of having a hematological malignancy, not for HIV. And there have been 6 other people who've had identical paradigms where they've had bone marrow transplants from a CCR5 negative donor for a hematological malignancy, and sadly all of these have died through complications of the malignancy or of the transplant. So it is certainly not something that could be rolled out widely to other people. So there are a number of different approaches to try and find ways to get round this. Therapeutic vaccines by themselves don't seem to have done much to help eradicate the latent load. There have been various approaches to try and wake up the latently infected cells, to try and reactivate the virus so the immune system can see them. And those using drugs which are known to switch on HIV, although they've been shown that they do that, you can detect HIV replication where you couldn't before in the appropriate cell populations. That doesn't seem to have had an impact on this latent reservoir. So those drugs alone haven't worked. So the vaccine by itself hasn't worked. The reactivation of the virus by itself hasn't worked. And that led us to the combination approach, so-called shock and kill or kick and kill, where you therapeutically vaccinate the person to try and enhance the immune response as much as possible, and then wake up the virus and hope that this primed immune response will be ready and waiting for every latent virus that reactivates to kill the cell off. But unfortunately, despite several studies being done, there's only been one that's shown any minor change in the latent reservoir. And the largest study, which we were involved in, the River study, the first ever randomized control study, which is just completed and will be coming out in the Lancet quite soon, again didn't show a depletion of the latent reservoir with pretty effective shock and kill treatment. So that as yet hasn't shown promise, but it's given us a lot of clues as to where we should go. We don't know what the next best treatment should be. There are better drugs to reactivate the virus, but there's evidence that different viruses may have different degrees of latency. So some may be more easy to wake up than others. And one would suspect that the easier ones to wake up will be the easier ones to eradicate, and then you may still be left with the more difficult ones to eradicate. And so it may need several rounds of shock and kill to get it to work. And then this becomes rather expensive and logistically testing, particularly for the poor individual who has to undergo all these treatments, where they could be quite happily taking their 1 tablet once a day and be perfectly well. There are other approaches to try and silence the virus that's in the person already. The catchy term is the block and lock approach, where you try and make the latent virus incapable of reactivation. And there are certain drug approaches which are being tried out to do this. That, to me, has always sounded like a good idea as a relatively short-term maneuver, but again, I worry that you'd be 100% effective at blocking and locking every single latent virus in an individual. And associated with this, of course, is what happens to somebody in whom you've given these treatments. So one of the very important parts of any clinical trial is discussing what you're doing with the patients. And when we were talking about the shock and kill approach with our patient representatives, we had an idea that perhaps you could shock and kill and reduce the reservoir down as far as possible so you couldn't detect any virus at all. And then the person could go away and come back for regular follow-ups, a little bit like cancer therapy, and see whether the virus has recurred. And we were disabused of this very vociferously by our patient representatives, who said that, well, as far as we're concerned, if you send us away not knowing whether the virus is going to reactivate between that appointment and the next appointment, it's all very fine being able to treat us when it does reactivate, but in the meantime we might have infected our partner or somebody else. So if you're going to have a block and lock or a shock and kill type approach, you've got to work out what your monitoring is going to be to make sure that you're not putting that person or their partner at risk in the meantime through inadvertent reactivation of virus.
Could a patient taking antiretroviral medication in the meantime almost give the impression that they have been cured, that the virus hasn't rebounded because they've been taking the the medication and its...
Well, yes, that's true. In fact, one of the issues with the patients who have been cured is the concern that perhaps they were surreptitiously taking the drugs anyway. So you can actually do therapeutic drug monitoring to test this out. I guess I would wonder why people would want to pretend they were cured unless they were trying to pretend to a partner that they were no longer infectious, which they could do anyway by taking the drugs.
And going back to, we've almost had the two different approaches involved in suppressing the virus's replication and almost counterintuitively boosting its replication. So how is it possible to actually activate a virus so that the immune system can recognise it?
Well, the what's called the proviral DNA, which is integrated into the chromosomes of the cell that the virus has infected, is just like a cellular gene, and it's got the same sort of structure with a promoter at one end and a polyadenylation signal at the other. And so it's switched on and switched off by the same sort of signals that our own genes are within our cells. Now, that means that if we know how to switch on our own genes, we should be able to switch on HIV. That in itself is a double-edged sword, because what you would like to do is very selectively switch on HIV and not switch on genes that you don't want to activate, because there could be all sorts of unpleasant consequences for that. So the approaches so far have been to target enzymes which are known to loosen up the DNA, the chromatin, and allow transcription of the virus to take place. And there is evidence that there are a particular class of these which are particularly involved in the control of HIV latency. So there is some degree of selectivity in being able to switch on HIV and not switch on other genes. I can't say we know for certain we're not having some sort of activating effect on other genes, but the drugs we've used so far are ones which have been licensed for other treatments in humans already, and therefore have got a safety side effect profile, which means we could justify using them in this. I think any new drug which came along, which we said was selective for HIV, would have to undergo very rigorous testing to make sure it wasn't switching on things we didn't want it to switch on.
So this is the so-called shock part of shock and kill. Moving on to the kill side, I assume we might have a similar problem or similar question to address, in that we don't necessarily want to activate all immune cells in a generic way.
No, we want to activate the ones which are very good at targeting HIV. And ideally, the specificity of the response is going to be determined by the vaccine that you give, so that you just will activate those which are specific for the antigens that HIV may present when it reactivates. One of the things that came out of the recent River trial was that the therapeutic vaccine certainly did boost the immune system, so that when we took cells out of people who'd been vaccinated, in the laboratory they were much better able to kill infected cells than before the vaccine. So that part of it is something we're probably better at than we are at waking up the virus. But again, the combination as yet doesn't seem to have been sufficiently effective to reactivate and eradicate cells.
And are there any prospects for gene editing, for example? I know that there's an idea that you could edit out CCR5, which you mentioned earlier, but are there any other ideas in the pipeline for this?
Lots. Gene editing is one that, as you mentioned, and of course there's been the famous case recently of the babies being gene edited in China to try and make them unable to catch HIV. That's acquired the appropriate response from the Chinese authorities. I think gene editing is something for the future. We don't know enough about gene editing and how specific it is and what sort of off-target effects might occur as yet. But if it's true, if we got very, very good at gene editing and we were able to either edit out CCR5, for example, so that the cells are non-infectious by HIV, or we were able to gene edit out the HIV provirus itself, that would be a very neat way of curing. Obviously, CCR5 itself is not something you would automatically want to take out of individuals unless there was a very good reason, because there are some consequences of being CCR5 negative.
So obviously HIV is just one virus, and there's been a huge amount of research into it. Has this translated into research into other viruses and potential treatments for a number of other viruses.
Yes, indeed. And hepatitis B has probably been the major beneficiary of this, in that it's also a sort of retrovirus. It's not quite a retrovirus, but it has the same sort of replication mechanism. And a number of the drugs which treat HIV are also very effective against hepatitis B. So that's one that's been benefited from it. I think that's probably the best example. People are now more confident about trying to treat chronic viral infections than they were. Hepatitis C is another one, but that's really a different sort of paradigm. But again, treatment for that has improved, I guess, encouraged by the successes in HIV. And now hepatitis C is effectively another treatable disease, in fact, a curable disease, which is something that wasn't thought of 15 or 20 years ago.
And this can radically change public perceptions of diseases, and we touched on stigma earlier. But what do you think, potentially, even if it's years in the future, a cure for HIV could do for the stigma surrounding the virus and being infected and living with HIV, even if actually your viral count is 0, you're completely asymptomatic, and you can't pass on the virus? It's still quite a burden for people.
It is. It depends where they are, what society they live in, and what the attitudes of that society are. It's very striking that in the areas where the education is poor, the stigma is greatest, and that's where the problem is at its worst. HIV is declining in virtually all parts of the world, apart from North Africa and the Russian Federation, and certainly a lot of that is associated with the stigmatization of people who are HIV positive or who undertake practices which puts them at risk of HIV. So one would hope that as education improves and people understand more, then the stigmatization should go away. All sexually transmitted diseases are associated with some degree of stigma, so I don't think we'll ever get rid of it entirely. Syphilis is just as stigmatized. In fact, in some ways, it's thought to be even more stigmatizing in a funny sort of way, and yet that's totally curable. So I think the correlation between curability and stigma is probably not absolute.
And are you hopeful for the prospects of curing HIV, for example, in the next few decades or 50 years? Would you? I don't want to press you for, say, a time frame, because I know it's very variable. But has enough progress been made that actually this is something that we could realistically hope for?
Yeah, you should never get into the habit of predicting things, because you're always wrong. It would be nice to say that things are going to be curable in the next 10 to 20 years, but I'd be falling into the trap that the Health Secretary of the United States did when they were making the announcement with Robert Gallo about the discovery of HIV as the cause of AIDS. I think eradicating latent virus is going to be a formidable task, apart from obviously the few occasions where something like a bone marrow transplant has taken place. But in someone who's infected, for which they have got reservoirs of infected cells hidden deep in places which antiretroviral drugs find it difficult to get to, such as in the brain or deep in lymphoid tissues, the chances of us being able to reactivate 100% of those to allow the immune system to kill off those infected cells is going to be, it's a big one. And as I said, the level of latency in different latent cells probably varies. So we might find it easy to deplete the reservoir down to a certain level, but then it's going to be incrementally more and more difficult to get rid of the more profoundly silenced viruses. And one wonders if you'll ever really get rid of all of them just by the sort of approaches we've been talking about. So I wouldn't be pessimistic, because things always turn up that we don't expect. And gene editing, for example, might suddenly become much more tractable, deliverable by safe vectors and cheap, that we can start winkling out each of the viruses from the individual cells. But I think 10 years, I think we will still be trying different ways of trying to eradicate latent virus. 20, I'm not going to guess.
Professor Leavitt, thank you so much for speaking with me.
You're very welcome.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think. Do leave us a review on Apple Podcasts if you have a moment, and you can also reach us via our website and our social media pages.